Yeah, we can go ahead and get started, and I will hopefully not completely butcher the name of our next presenter, who I'm very pleased to be able to introduce, Dr. Sadegh Shokatian. Is that good?
Close enough, yeah.
Okay. He's here today from Terramera, where he's the Director of Chemistry and Bioinformatics. He is an accomplished chemist with a focus in experimental and computational spectroscopy, and he leads a team of chemists and machine learning engineers in the discovery and optimization of Terramera's revolutionary ActiGate platform, which increases the performance consistency of active ingredients by delivering them directly into target cells. His experience includes over 7 years of research in the soft X-ray beamline at Canadian Light Source, and he holds a PhD in chemistry from the University of Saskatchewan and a master's in analytical chemistry from America University of Technology. And as Sam said, he is here to talk to us today about the discovery of synergistic combinations using machine learning. So with that, please take it away.
Great, thanks a lot for the introduction. So let me share my screen. Okay, can everyone see my screen?
Yes.
Perfect. So, as mentioned, I'm going to discuss the discovery of synergistic combinations using machine learning and a bit of computational chemistry. But before starting, I just wanted to introduce the company that I work with, because we're not in the pharmaceutical industry, we're in the agtech sector, and it might be kind of a question mark for you guys on why we're presenting here. But I want to show you why and how it's related to the drug discovery. So I'll start with the core question that we have. How can we use technology to unlock the intelligence in nature to ensure an Earth that thrives and provides for everyone? And based on that core question, we have three main BHAGs. To reduce the global synthetic pesticide load by 80%, to increase the global farm productivity by 20%, and to increase the soil carbon by 100%. And these are our BHAGs for 2030. So having those BHAGs in mind, we've gone ahead and developed two main platforms on our end. One is called ActiGate engine or ActiGate technology that I'll be talking mainly on that today. And the other one is the plant intelligence engine, which covers most of the machine learning, artificial intelligence, stats, data science, and data collections. So I'll just touch briefly on the plant intelligence engine just to show the capabilities of Terramera, and then I'll go over the ActiGate. On the PI or plant intelligence engine, we have the data collection on one side, which we want to use to increase the plant health and yield. We're using a universe of active ingredients that we have, so pesticides, insecticides, herbicides, different approaches that we want to take. We test the ingredients with the ActiGate technology. We screen for the pest and disease controls and plant health, and we collect those data, use them to train our machine learning models to be able to predict disease of the plants in earlier stages. So the idea is if we can detect the disease in earlier stages, say a week in or 2 weeks in when we're seeding plants, we can stop the experiments and go ahead with a new experiment. And that's the main idea behind the plant intelligence engine. Now, as you see, I'm talking about the ActiGate technology or ActiGate chemicals here. Just to give you an explanation on that, ActiGates are a suite of chemicals, a group of chemicals from different classes of chemistries that will enhance the performance of active ingredients. The way that we're doing that is by reducing the dose required for the active ingredients to be applied on the plants. And I'll explain a little bit more on that. So again, on the plant intelligence engine, which covers mostly our engineering department, we have four main categories. So on the software side, we've gone ahead and developed a digital lab notebook, which records pretty much all of our experimental measurements. We have a material synergy system that acts as a database that connects to our digital lab notebooks, and it's kind of an inventory system for our ActiGates. And we have a lab inspection app. On the robotics side, we've touched some of the mechanical work, such as developing the sprayers, as well as a lot of IoT work on bedbug detectors or terrameters, as we call them, which are the environmental IoT devices that we have. And we've just started working on sensor packs for field imaging. On the computer vision side, we've done a whole bunch of works on plant disease estimations, various counting such as flower, fruits, cells, nematodes, and aphids, cabbage liver weighing, and new work such as non-destructive sugar content estimations of wine grapes. And on the stats side, we've developed a sophisticated statistical pipeline that takes all of the experimental data and performs a whole bunch of stat measurements on these data. So enough about Pi. Going to the Actigate discovery pipeline that we have, the idea is that we have some in vitro, in plant, and in field measurements done by our biology team. We use those data to train our machine learning models and come up with hundreds or thousands of molecules as predicted molecules that can have synergy with our active ingredients. And then we take those chemicals into the computational chemistry side and try to filter them out to reduce the number of predicted activates, and then those ones will be fed to the experimental measurement. So this way we're reducing the experimental costs since they are expensive. Now what we've done so far on the Actigate discovery pipeline, we're counting them as two pieces, computational chemistry and machine learning piece. On the computational chemistry, simple works such as quantum calculations of log P, free energy of complexation, dipole moment, surface area, polarizability. And on the classical approach, free energy of solvation, membrane permeability, co-solvent selectors, CMC predictors, and enhanced configurational measurements. And on the machine learning side, we're taking both predictive and generative approaches. So to predict molecules for a given properties using the existing data sets, and to generate new molecules using a given property of interest. So I've talked about synergy a little bit. Just to mention what synergy means, we have different definitions for synergy. Just to mention a few of them, the highest single agent saying if one of the active ingredients has a synergy of 1 or efficacy of 1 and the other one is not active, when they show a higher synergy or higher efficacy when we're adding them together, it counts as a synergy. Or response additivity saying the efficacy of 1 plus 1 is greater than 2. This is pretty much similar to the response additivity with the difference in the probability. And the one that we've taken, and I know that it's been used in pharma industry as well a bit, is low additivity, meaning that we're trying to achieve the same degree of efficacy using a lower dose of the active ingredients. So we've taken that. We've looked at the chemical space map that we're working with. So this one is just two visualizations of the chemical space map that we're working with, using the zinc 250K as the baseline, having 250,000 molecules. Here I've compared them with the known fungicides in this case and potential activates that we can deal with, and only one of the generations of activates that we have. And as you can see, we've covered just a small piece of this space. It can be observed on both cases. So there's a lot of potential for us to go through the discovery of these chemicals. So what we've done from this part, I'll be a little bit more technical, just so you guys know. So what we did, we started with a public data set. We extracted roughly 25 million molecules from that data set. We tried to filter them out using things such as toxicity of molecules or heavy elements, or if we have metals. So these molecules were all eliminated. And then we used a similarity filter, which I've shown on the left side, just to show you what it is. So say we have a random molecule, fructose, a very similar molecule to that will be glucose. And I've shown a kind of a similar molecule and a very different molecule. So these are done using Jacquard similarity indexes, if you guys are interested in knowing. Using those similarity filters, we reduced the number of our prediction data set to 500,000 and divided that into groups of just 5,000 molecules for the predictions. So then the next logical step was to put them in a neural network. So we have the features from our active ingredients, the features from our activate. Let's just put it in the neural network and see what happens. And with that, we can come up with a binary prediction of yes or no synergy. So when we did that and we figured that we were achieving an accuracy of roughly 70%, we decided to increase the performance of our model. So we designed a neural network ensemble that gives us a distribution of synergy rather than just a binary. So this way, we can have the probability of synergy, we can have the confidence interval with a fewer false positive and a lot more data efficient. The next step for us, which we've just started recently, is to implement an active learning approach. So the idea is to divide the data set into clusters of molecules that you can pick the most representative and the most informative molecule from each cluster, put them in the HTTP screening, and use those data to increase the accuracy of the model. In here, I've shown an example of the differences between random selection of the data with the active learning approach. And as you can see, using the active learning approach, since we're picking the most representative and the most informative data, we can achieve a lot higher accuracy with a lot less labeled data. So that's all about the predictive side of things. We recently started working on the molecular generation. So the idea is we're exploring a space of 10 to the 60 molecules, which is essentially more than all of the atoms in the universe. And we're just exploring just a teeny tiny bit of this space, roughly about 1 billion molecules. About 10 to the 50 of these molecules are not even discovered yet. So there's a huge potential in exploring this space. That's why we stepped into the molecular generation models. So using different generative models, we're exploring this chemical space based on the features that we have in mind. So in this case, synergy or efficacy. And we're generating new molecules that are first theoretical, so not discovered yet. Second, have a huge potential in IP for us. And we're expanding our IP using this approach. So just to give you a few examples on the works that we've done after we've done the predictions. Here I've shown one of the examples of finding something called on our end, something called the enhanced configurational sampling. We're using the multi-replica MD plus configurational exchange and metadynamics to pick a few configurations for the samples. We go to neural replica and then a bit of clustering to come up with the winner molecule that has the lowest energy configuration using a classical approach. Oops, sorry. Hope it goes back. Oh, perfect. So in here, I've shown some of the examples of the works that we've done on the ComChem. On the left side, I don't know why the movies are not working. You can see some of the modules of the enhanced configurational sampling. So we're basically putting the molecules, the active and activate, close to each other. We're studying how the energy changes when we're changing these configurations. We end up with plots of the energies and pick the molecules with the lowest energy geometry. On the top middle, you can see the interface studies that we're doing. So we have water molecules in both sides. We're putting some of the selected activate molecules in between, and we're studying the changes in the energy and the orientations of molecules in the interface. Interestingly, we've picked some of the really good activates that we have and the bad ones from the same class of chemicals. When we've studied them, we've seen a huge difference between the orientations of the molecules right in the interface. On the bottom, you can see one of the interesting works that we did with the exosec technology, which is essentially a new method of delivery offered by the company called Exosec that was purchased by Terramera. We're putting our active and activate ingredients inside the a wax nanoparticle, all of them putting them inside a nanoparticle of water or non-droplet of water, and in interaction with a hydrophobic surface. So we realized that using this approach, using this exosec technology, our activates tend to get out of the water and start interacting with the hydrophobic surface. Now, the interesting part is when you run this simulation without that wax in between, you don't see these kind of interactions. So the activates will stay in water and they won't interact with the hydrophobic surface. And on the right side, you can see one of the core pieces to our activate discovery is the membrane permeability. So based on the molecules that we've picked from the machine learning approach. We're going to put them together with an active molecule, with an active ingredient outside the membrane. We push them through the membrane, and we study the changes of the energy through the membrane. And that led to some really interesting results on the way that we thought about the mode of action and the interactions between the active and activate. But as you can see in here, so the green blob is the membrane, top side is the outside of the membrane, and bottom is inside of the membrane. The red molecule is active and the blue is activate. And as you can see, the activate molecule pushes or pulls the active ingredients into the membrane. They go through it, and at the end, the active goes out, and the activate stays inside the membrane and interacts with the membrane molecule. The next piece was the interactions between the different types of interactions between active and activate inside water. So when we have these molecules, we have different types of hydrophobic, hydrophilic interactions. And we studied these interactions as a function of, we studied this free energy of calculation as we have them together. The next piece is the study of these active and activate molecules together at low pH and high pH. So we understood that in this particular case, which is not an activate, by the way, in this particular case, we can see the aggregations of molecules at low pH, and in the high pH, we can see that they just like to expand throughout the unit cell. So with that in mind, we've tried to modularize everything that we have from the machine learning ones all the way to the computational chemistry ones. We're in collaboration with Microsoft and trying to convert these ones into Dockerized containers that we can share with people if we want in future. It's one of the business models that we have in our end. With that, I try to speak super fast to have more time for questions if there's any.
Thank you again, Dr. Sadegh, for your presentation. And just a personal question from my end. I'm just wondering, are you familiar with a company called Gevo from the West Coast, Gevo?
Gevo, I'm not.
Okay, I was just wondering because
yeah.
Your first initial slides, I don't know, it might be a company that you may want to look into based on your background. And yeah, just a remark from my end.
Thanks.
So thanks for a really great presentation. It was very interesting. I'm not sure that I followed every part of it, given it's not my field, but I did find it very interesting nonetheless. And I have perhaps a naive question to ask. You mentioned that there's 10 to the power of 60 potential molecules, and so I guess my question is, how do you actually know where to start with that many molecules to begin with, and then how many can you actually assess within your technology system at once?
Mm-hmm. That's actually a great question. So what we're starting with to train our model is to use all of the existing molecules out in the market. So roughly about 1 billion molecules, organic molecules, are discovered so far. We can take all of those molecules, try to generate a chemical space, and work on that space. So depending on the features that I want, say as an example, log-P, which is known in the pharma industry. So I'm taking a log-P, a molecule with a known log-P on that space. I'm working a little bit further from that, and I'll try to generate a molecule that represents a point next to that particular molecule. So that way, we can define the steps, the gaps in the steps that we want, and get further and further from the given properties to generate new molecules.
Okay, thank you.
Thank you again, Dr. Sadegh, for your presentation. And I guess we will be going, we have 2 more presentations left before we go on our lunch networking break. So if we can all go to the next room, which is Data-Driven Workflow Optimization by Ms. Fleur Lenda. So if we can all just go there.
Would like someone put their hand up, Sam?
Oh, your hand up?
Yeah. There's a question now in the chat.
It's not letting me hover my mouse over there. I think my screen is frozen, unfortunately.
Do you want me to ask it then, Sam?
Sure, yes, please.
Okay. So this comes from Marwan Ahmed, and he says, he or she, I'm not going to guess actually, great presentation. For the pH simulation, do you use constant pH MD simulations, or you start from a protonated system at the specified pH and run traditional classical simulations afterwards? Which technology do you use of molecular generations, GANs, VAE, etc.? And do you drive your molecules towards the desired property space or filter afterward?
So for the first question, yes, we start with the protonated system and then run the classical simulations on them. For the generative models, we're actually trying both approaches, both adversarial approaches as well as VAEs. We're trying to improve the accuracy and see which approach gives us better results on that end. And the third one was, do you drive your molecules toward the desired property space or filter afterward? Yes, that's exactly what we're doing.
Great.
Okay, great.
Thank you so much again, Dr. Sadegh, for your time. And we'll be heading to the next presentation, which is data-driven workflow optimization.